The next experiment you will be doing relates to chromatography. Chromatography is a general concept of separation of different compounds using a variety of methods. Sometimes this involves using light. That's why the beginning of the word chromatography, chromo, relates to light. However, this is not the only kind of technique that you can use. So we'll be looking at two different kinds of chromatography. One is column chromatography. The other is thin layer chromatography, or TLC. So the basics of chromatography, as we will use them in this lab, is that there is a solid material, either silica or alumina, that provides a relationship to a solvent that has the material you are separating dissolved in it. We are playing with intermolecular forces here. A very polar solid material will attract more polar materials. That means that nonpolar materials will travel further on a solid material, whether you are using column chromatography or thin layer chromatography. So column chromatography you can do either very small scale. The way that we usually use this in lab is you use a Pasteur pipette that can either be about 6 inches long or 9 inches long. You put in a plug of cotton and you fill it up with alumina or silica gel, and then you dissolve the material to be separated in some kind of solvent, generally nonpolar to start off with. You put it on the column, and then it runs down the column, and the different intermolecular forces will allow the different parts of your mixture to separate. The reason why we do this on extract of spinach is that the materials that you are separating are very easily visible. Chlorophylls are green, carotenoids are orange, some of the other materials can be blue, green, or gray, and you really can see the difference in those colors using our variety of separation techniques. So in the column, you would see two different bands. You would see a yellow band of the less polar material coming down first. You might change solvents, and then you would see the more polar materials start coming down, and then you can separate them into different flasks. So that is the nature of column chromatography, to be able to separate different materials using an eluent, and the eluent is the solvent, and then collecting them in different flasks and removing the solvent and isolating them that way. So this is a very common technique in organic chemistry. In order to do some very quick analysis, you can then take those separated materials and use something like TLC in order to see the different spots. So the TLC plate, generally a rectangle, the base material is either foil or plastic, and then it has silicon gel embedded on it. You spot a little bit of a solution of the things that you are separating onto the TLC plate. You place it in a chamber, and very similar to capillary action, the solvent will move up the TLC plate. You may have tried a technique like this with paper and markers. So if you spot on the bottom of a piece of paper, say black marker, and then put it in water, the water will creep up the paper and you will start to see the separation of the pigments. So it's a very basic way. If you want to try it out, it's a very basic way to see the technique that we're doing with TLC. In this case, you'll see different parts of your spinach extract going up the TLC plate. And in the case of the green spot and the crude, you're going to see all the individual pigments be separated out, and you can actually see them on the TLC plate. So these are two different methods to be able to analyze and, practically speaking, separate different materials from a mixture.